Now we are starting with the topic planning of experiment. The planning of experiment includes the different practical steps which is needed for planning and conducting an experiment. It includes the following 7 major steps. The first one is we need to recognize and determine the objective or goal of that experiment. Then we need to determine the response variables or the factors that is generally causing the variation in the experiment, and we need to measure that particular variable. Then third steps, we need to identify the factors which are varying and in both the test as well as in the control experiment, and which factors has to be held at constant or its influences should be minimized. Then we need to make a selection of the response variable. If we are performing different factors for an or if we are analyzing different factors for an experiment, what are the response or what type of factors that we need to select for this response variability. Then after that we need to choose a design for an experiment. And after designing experiment, I have told you what about the designing of and how can we design an experiment by means of various techniques. And after the designing is over, we need to conduct the experiment. After conducting the experiment, we need to determine the number of replications or repetitions on the basis of experiment to conduct. And this replication, we know that we are generally performing this replication for ensuring the acceptability or the actually the core value of that experiment. Then after the replication of that experiment, we need to take the analysis, that is the statistical analysis of the experiment, and based upon the statistical analysis we need to draw the conclusions and making recommendation in response to that experiment. So these are the different ways that we need to plan an experiment. Then while we are doing this planning, we need to follow some procedures. What are they? We need to keep everything unchanged except for a single variable, and this single variable should be subjected for the testing. And a control group should be used when conducting an experiment. These groups receive the same attention as the test group. Okay, so these are the different procedures or different steps that we need to keep in mind for doing for planning an experiment. Then I will explain this planning of an experiment by using a suitable example. If we are observing, I told you in the previous class how we are conducting an experiment, what are the scientific ways to schematic representation of a scientific method. We are starting with the observation and defining a problem, and then we are asking questions, and we make a hypothesis just like that. So here we are starting this or planning this experiment by asking a question to that experiment. For example, does the plant extract A has the property to kill the mosquito larvae? This is our question for a particular experiment. And based upon this question, we are making a hypothesis that at certain dosage, the plant extract A will causes 100% mortality of mosquito larvae. So here, what is the factor or what is the variable that we are taking in or we are planning for this experiment? Yes, a particular dosage of the plant extract. And what we are expecting, it will causes mortality or death to the mosquito larvae. Okay, so based upon this two analysis, we are making the hypothesis that at a certain dosage, this plant extract A will causes 100% mortality to the mosquito larvae. So what is the variable here? The plant extract. The plant extract at various doses or various concentrations are the variable which is generally performed in the test sample. Okay, then while we are doing this experiment, something or some factors has to be made constant. What are they? That is called the test constant. All mosquito larvae that we are taken for analysis should belong to the same species. Then the developmental stage of all the larvae should be the same. The number of larvae which we are performing in all tests and also the control should be should be constant. Then the water that we are used for this method should be distilled and its source should be the same. The quantity of water should be the same in all experiment or all samples, and we are exposing this experiment for about 24 hours. So this will be same in all samples. Then we have to set up a control group. What is meant by a control group is that this group will be similar to that of the test, but it is except it does not have any plant extract. This control group contains only the water and the mosquito larvae and all the other conditions, but not the plant extract. This is called the control. Okay. Then after the experiment is over, we are analyzing the data. I told you that in the steps we have keeping this experiment for about 24 hours. So we are observing this experiment after 24 hours is over and note down the number of larvae which is died at various doses and in the control. Then here we are calculating the percentage of mortality of mosquito larvae. Based upon this analysis, we are making a conclusion that did larvae die with the plant extract A? This is one of the conclusion. And what was the lowest dosage that is producing 100% mortality to this particular mosquito larvae? So that's all about the conclusion. We are making the conclusion based upon this analysis as that is there any effect to this mosquito larvae by applying this plant extract, and what about the, if it is yes, what about the lowest dosage that we can be applied for making 100% mortality to the mosquito larvae. So this is the planning of experiment. Okay, in this way we are planning an experiment. The main purpose of this planning of experiment is we can reduce many resources like time and energy, all these things that we can be reduced by the effective planning of an experiment. Okay, so that's all about the planning of an experiment. Thank you.